Hello everyone, my name is Annabel Klemets. I'm a second-year PhD student and a junior research fellow at the Institute of Genomics, University of Tartu, Estonia. And today I'm very happy to present the results of our new preprint titled Fecal Microbiome Predicts Treatment Response After the Initiation of Semaglutide or empagliflozin uptake. So first I will briefly introduce the importance of this topic. So many medications are known to extensively affect the gut microbiome, its composition and diversity. And there is already strong evidence for anti-diabetic medications as well. For example, for metformin, which is used for treating type 2 diabetes. But we still don't know exactly how to other anti-diabetic medications like semaglutide or empagliflozin affect the gut microbiome and vice versa. And that has very important consequences in clinical practice because outcomes for patients at the end of type 2 diabetes therapy can vary very greatly because of that. And for this exact reason, we initiated our study. So we invited 20 patients with type 2 diabetes into our study that matched the following inclusion and exclusion criteria and asked them to donate a fecal microbiome sample and a clinical blood and urine samples before the treatment at baseline and 3 times during the treatment period for a total of 12-month period. We performed 16S gene analysis for the microbiome samples and analyzed common treatment markers from blood and urine samples. And we were mostly interested to see what were the bidirectional interactions between the medications and the gut microbiome according to these 2 study questions. So first, we were interested to see what is the effect of semaglutide and empagliflozin on the gut microbiome. And we started out by analyzing gut microbial diversity because many other medications have been shown to extensively affect the gut microbiome overall. So we saw by analyzing alpha and beta diversity that the changes mostly occurred later during the treatment period, not between the baseline and the other time points. And the changes were overall mostly individual and quite moderate. And in addition to that, we also investigated changes in the bacterial abundance in the gut microbiome. So here on this figure are shown changes of the most significant bacterial genera and their CLR values in the semaglutide study group. And we can see that the changes also, similarly to the gut diversity, occur later during the study period and depend also on the genera as well. So we can conclude from that semaglutide and empagliflozin use seem to mostly affect the compositional values of bacterial genera in the gut microbiome, not so much the gut diversity itself. So in the second part of the analysis, we were interested to see if we can use baseline gut microbiome data to predict how well do patients with type 2 diabetes respond to the treatment by using the most important major clinical treatment markers. And we correlated the values of baseline gut microbiome before treatment with the changes in glycohemoglobin values by the 3rd month. And glycohemoglobin is a very important treatment marker for type 2 diabetes because it shows the changes in blood glucose levels over a longer period of time. And indeed, we saw that multiple bacterial genera and their compositional values and other metrics as well were associated significantly with the changes in glycohemoglobin levels by the 3rd month in both study groups. So to sum up our most important results, we revealed that there are changes in the gut microbiome following medication use, although they are mostly indirect, possibly due to the overall effect of those medications following treatment. But however, most importantly, baseline gut microbiome indeed was associated with a change in major treatment markers like glycohemoglobin, for example. And our results definitely deserve further investigation for understanding better the potential causal role and functional significance. So with that, I would like to thank you, my wonderful co-authors, and also our collaborators from the University of Tartu Hospital. Thank you very much for listening.